Hello, everyone. My name is Soumya Iyer, and I would like to share my short presentation of my final project for the deep learning course. So I would like to start out by jumping right into the motivations for this project. So currently, response to cancer therapies is widely variable. Extracting information about cancer therapies from clinical trials, it's expensive, it's sparse, and non-uniform. Cell line models, which we have developed in our group at the Broad Institute, they're a useful proxy to perform high throughput screens of drug response. They're scalable to thousands of samples across hundreds of drugs. And each cell line is molecularly characterized, and the data is structured and is uniformly measured, making it an ideal candidate for machine learning. The problem statement that we have is, given molecular characterizations of these cell lines, can we use deep learning methods to model response of each cell line to cancer drugs? And it turns out we can. After applying the model, we saw R-squared results that are on par with or better than currently what's out there in literature. So you can see here that across many drug pathways, there's variability across folds and seeds, etc. So even within the same pathway, we have variability between drugs. So drugs targeting the same gene, for instance, have different R-squared, different predictabilities. This is another example of the same thing where you have cell cycle as a pathway, and each drug responds differently because of differing mechanisms. So given that, there's more to do clearly in this, and this is just a starting point, a decent starting point. The limitations of this are limited interpretability due to latent representation. So if I want to find out what contributed, which gene contributed to a particular output, that's hard to do now. I cannot predict new drugs, only the ones that have been screened, and gene or mutation level importance is difficult to extract, which is similar to the first point. So I thank everybody for listening, and thank you to the staff for their help throughout the course. Thanks.